and then protein processing as well. So we, last time we ended off with how do we get the genes transcribed? So, right, we have our RNA. Well, what's happening with the RNA? So that's what we're going to talk about first. And what's happening with the RNA is really important for the protein that's encoded by that RNA. So we're going to talk about that. We'll talk about when the RNA can be processed, protein can be processed. Right, and always keep in mind that one of the most important concepts of understanding cell biology, there's a lot of different cells. So how do, in our body, right, over 200 types of cells, how do they become different? So we talked about differential transcription, but there can be differences in the RNA as well. And so that's kind of the crux of alternative splicing. How you go from a single gene that encodes a single kind of pre-mRNA, where you can get a ton of different proteins according to different patterns of it being spliced. So that's super sweet. So we'll talk about that. And then I alluded to before the fact that the RNA transcript gets processed. Right, so it gets at the 5' end, it gets the cap, and at the 3' end, it has this poly A tail. And those serve to protect the transcript, but they also help. They help with getting the transcript exported from the nucleus. They help in regulating translation. So they're important for a lot of reasons. So we're going to talk about that in detail today, and talk a little bit about selective degradation of RNA. So what happens with the RNA that isn't processed appropriately, or the RNAs we don't want? They are degraded, and so we'll talk about that as well. All right. Oh, it's so good. Okay. Oh, it's not wrong to love it. I admit it. Post-transcriptional RNA modification. So co and post, right? So co-transcriptional RNA modification implies that modification is happening during transcription, and then post would be after. Right, and so I kind of lump them into two, and that should become clear as we go. All right, so in this image, we have our DNA that's being transcribed, and the primary RNA transcript, which, right, there's a lot of terms for this. Primary transcript, nuclear RNA, pre-mRNA. It basically means the unspliced version of the RNA. So this is the version of the RNA that still has all its introns. And this transcript gets processed. Actually, this image isn't totally correct, because this makes it look like the whole thing is made and then the cap's added. The cap's actually added kind of right away. So when RNA Pol II starts to transcribe a gene, it gets it's estimated about 25 nucleotides in, and that cap is added, right? And that's partly because you want to protect that RNA right off the bat. So in the nucleus, there's a bunch of enzymes called exonucleases. Write that out. We'll talk about exonucleases. They're both our friend or our enemy, right? We need them, but sometimes we don't want them to degrade things. We want them to degrade things, the right things at the right time. And if we are just transcribing a new RNA that we want to ultimately get translated, we don't want an exonuclease to degrade it. So first of all, exonuclease. All right, so it has the ASE. We know it's an enzyme. It chews up nucleic acids. Exo from the outside, right? So if we have our RNA 5' to 3', and we'll call this our RNA. There are exonucleases that will chew from the outside in. These would be exonucleases. This, because it's chewing in the 5' to 3' direction, is called 5' to 3' exonuclease. This, chewing the other way. So this is referred to as a 3' to 5' exonuclease. So again, exonucleases big old gene family. It's a ton of different nucleases, and they're chewing up the nucleotides in a little bit different way. So in this case, this has affinity for 3' end, this has affinity for 5'. Exo chewing from outside. That's different from endonucleases, right? Just kind of attacks a sequence in the middle. Have you guys ever heard of restriction enzymes like GecoR1? Those are types of endonucleases, and they're really specific for the exact sequence that they're going to chew up. We actually I don't think I talk too much about endonucleases in this class. I talk a lot about exonucleases, but for comparison, I think it's important to get the difference. Okay. So there are a bunch of these nucleases in the nucleus, because there's a lot of RNAs we want them to degrade there. We don't want them degrading our new transcript, right? So right when an RNA is produced, that 5' end is the first part that's produced, the 5' cap is added right away. And that's to help protect it from getting chewed up, right? So this image, I get why they draw these in textbooks. I get why their intention for doing this. But I just want you to understand, this is a little bit misleading because the cap is there. It's added right away. Okay? All right. And then actually as the RNA, yeah, question?
So the exonucleases are there to eat up the whole RNA that they really do?
Yeah, they serve a really important purpose to chew up RNA that you don't want in there. For example, when we do splicing, we're going to splice out the intronic RNA. We don't want to have a whole the nucleus would become like a garbage dump for intronic RNA if we didn't have something chewing it up and breaking it down. So exonucleases are really important there to chew up that intronic RNA that gets removed. So they serve a really important purpose. It's just the cell has mechanisms to make sure they're chewing up the RNA we don't want rather than the RNA that we do want. So splicing actually starts to happen as transcription is occurring as well. So that is regarded as a co-transcriptional RNA modification. And then the poly-A tail, right, that's the last thing that happens. It's the 3' at the 3' end, and the addition of the poly-A tail is actually what terminates transcription. Okay. So those are the modifications that we're going to go into detail on right now. So again, 5' capping. I'm saying that with intention because I had a lecture where I just kept saying 5' crapping, and I couldn't stop. Like every time I said 5' cap, I said 5' crap, and it's, oh. Okay, so I worked really hard now to say capping. 5' capping, it's most commonly referred to as the 5' cap. The cap is sometimes referred to as the 7-methyl G cap or 7-methyl guanosine cap. So the longer the name, the more descriptive it is about kind of the what the cap, the molecular nature of the cap. So it actually is a methylated guanine nucleotide that gets added in a special way. It gets added kind of backwards. So we'll talk about that in a second. 3' poly-A tail, right? So it's poly many and it varies. On average, there's like 150 to 250 adenines that are added to the 3' end of the RNA. A big old poly-A tail. It's going to vary in length. We'll talk about the ramifications of that variation. And then, right, what is super sweet, those are important too, and those are cool, but what's really sweet, right, is how you get differential splicing, right? So we can, from the same gene, get a bunch of different protein products, right? So does anyone know what the record is? From a single gene, how many protein products can you get? It's in this lecture. It's DISCAM, and it's over 38,000. 38,000 different predicted peptides, and so so software predicted it based on how many introns and exons it had, and then scientists at the bench went and had to confirm, are those actually all expressed? And over 20,000 of those have been confirmed. So one gene has the potential to produce more proteins than we even have genes, right? Because we have about 24,000 genes in our genome, 22 to 24,000. Crazy. So you can imagine like how specialized your cells can become if you can create that much variability in the proteins that are produced by a single gene. Mind's blown, right? Mind's blown? Yeah? Okay. Fantastic. So before I go on, I think when I used to give this lecture, I would see a lot of confused looks when I started to talk about, bless you, why the 5' and 3' UTR are both important for translation, right? Because you think, well, usually it's drawn like this. We have our 5' end, and that's where the ribosome forms, and then it, slides along, finds the first AUG, and just keeps going, and it's this linear thing, right? Well, it turns out that especially mammalian messenger RNAs are actually in a circular conformation, right? So that's what this image is showing. In blue is our mRNA. Here's the 5' cap, right? And the cap is surrounded by proteins, and we'll talk a lot about that. Here's the poly A tail. So check it out. Instead of poly A tail being over here like sad and lonely, and just I'm not doing anything else to do at the beginning of translation, it's associated with where translation initiates and is really important for the initiation of translation. So that's why here's our 5' end, our 3' end, they both matter for initiation of translation occurring properly. In fact, you need that circular conformation in order for translation to be initiated. All right. And the protein here that you might be going, what the heck is that? EIF. EIF stands for eukaryotic initiation factor, right? And so in eukaryotic cells, this is a group of proteins that are important for the initiation of translation, specifically. These exist in the cytoplasm. They're not nuclear. These are in the cytosol. So these associate with the transcript once it makes it out of the nucleus, and they help initiate translation in the cytosol. You got it. I have this, this same image is later in the lecture. I need to give this preview, I think, to give context to some other things that I talked about, so I won't lose you guys. So we'll go over those EIFs again. Scaffolding your learning to make sure I don't lose you guys. Okay. Questions so far? No, just pumped to learn more. Okay. So, so let's talk about the 5' cap first, because it's what happens first. And so prokaryotic mRNA is largely unmodified. So it doesn't have this 5' cap. And in fact, when the transcripts are produced, right, they're in the nucleus, or they don't have a nucleus, right? It's just in the cytoplasm, and so it doesn't need all of these special mechanisms to get it out of the nucleus. It's just there, and the translation machinery is there, and oftentimes you get multiple proteins produced from kind of the same RNA. So eukaryotic RNA, different, right? We're more complex. Eukaryotic mRNA is capped and polyadenylated, and so the cap happens at the 5' end, and it is referred to as, right, I mentioned 5' cap, that's what I'm going to say, but if you ever see 5' 7 methyl guanosine cap, it's the same thing, it's just, I don't know, someone wants to sound smarter, all these extra syllables, or more descriptive, right? So it is a modified guanosine that has a methyl group on I think actually the nitrogen is at position 7, hence the name 7-methylguanosine cap. I'm not going to ask you to tell me that on the test. Okay. But I know I have some biochemists in the room, and they might appreciate that information. Okay, so here's a better look at the structure of the 5' cap. And so I mentioned that it's the way that this modified guanosine is added is a little bit funky. So if you look at the RNA, so RNA, just like DNA, the nucleotides are added 5' to 3'. The 5' to 3' refers to the carbon, the position of the carbon, right? And so here, here would be the 5' carbon, and it's connected to the 3' carbon. So 5' to 3', 5' to 3'. That was in crap, but right over and over. So the way that the 7-methylguanosine is added is by a 5' to 5' linkage. So that's abnormal. 5' to 5'. What did I say this exonuclease recognizes? A 5' to 3'. It doesn't recognize, because of the difference in the way that's added, that creates a different structural conformation here that 5' to 3' exonucleases don't recognize. Does that make sense? Okay. And so the way this is added is there are 3 different enzymes that add it. I am not telling, you guys can, if you guys go through the PowerPoint lecture, you notice sometimes under the slides there's a bunch of text. I've just grabbed text from the textbook usually, or from primary papers, to give you more details if you want them. So if you want to read about the 3 different enzymes that add this on, you're welcome to do it. This lecture is so dense already. I kind of, I want you to understand that this is added by enzymes, but I'm not testing you on the names of the specific enzymes and how they're adding that. Right, so I'm always trying to strike the balance of giving you kind of the right level of information, and if students want more, I can give it to you. I can give you primary papers if you want. I can make this as hard as you want, but right now, what I want you to appreciate is this cap is added, 5' to 5' linkage, that protects this new transcript from those exonucleases. They don't recognize it, okay? So that's how it protects it. It prevents the exonuclease from recognizing it. That cap isn't there, the nuclease is like nom nom nom, Cookie Monster. Yeah, okay. So that's not the only thing the cap does, because what happens when the cap is added, there's a protein that is very nicely named by the nomenclature people, cap binding complex. Right? CBC, cap binding complex. It's named that because it binds the cap. And it also is really important for performing the functions that are associated with the cap. So here's our laundry list of functions that the cap is important for. So the cap, when it binds this cap binding complex, it helps recruit the spliceosome that removes the first intron. So it's important for removal of that first intron in any transcript. So they've done experiments where what they've done is they've prevented addition of the cap and then observed what happens. And one thing that they noticed is splicing didn't occur normally. And it tends to be just the first intron isn't removed. For some unknown reason, it doesn't seem to be important for splicing of subsequent introns, but it is important for splicing of that first initial intron. Okay. Regulation of nuclear export. So the cap binding complex helps associate with the nuclear pore to help get that transcript out of there. It's not the only thing that's required, but it's one of the things. It also helps translation occur in cytosol. So you might have noticed in that first slide, the 5' cap. It's so hard. 3 letters. The 5' cap, there were EIS bound to it, right? You didn't see CBC there. But the way those EIS get there, the cap binding complex recruits them. So when the transcript that has cap binding complex that helps associate with the nuclear pore, helps get that transcript into the cytosol, it then is like, hey, eukaryotic initiation factors over here, recruits them, and then it takes heights, like my work is done. But that's how EIS get there. So if you remove the cap binding complex, EIS have no idea where to land. Right? They don't get recruited. So that's how it promotes. The cap is important for translation in the cytosol. Association with the cap binding complex, right? The cap binding complex recruits those EIS, and then the EIS bind directly to the cap. Okay. And then this one I kind of already went over. Because of this 5' to 5' linkage, exonucleases don't recognize it. All right. Good. Okay. Cool. Right, and the way that all of this is figured out, what scientists do is they're like, okay, there's this cap thing. What's it do? You mess with it, right? That's what scientists do. We mess with things and see what happens. So they remove. You can do all sorts of things, like maybe mess with one of the enzymes that's responsible for adding the cap. So now the cap doesn't get added. What happens? What happens is splicing gets messed up. Export doesn't happen. If you somehow put that were able to get that transcript to the cytosol, it would be translated, and it would get chewed up by exonucleases. Okay. So poly A tail, how's that going? The poly A tail. So first, it's this long stretch of adenines at the end of the transcript. All right, so the mRNA, the pre-mRNA, basically what it has are binding sites for the machinery that add the poly A tail. Okay, so what I mean by that, I wish I had more whiteboard space, but I don't. Okay, well, let's talk through it first, and once I go through the slide, I'll do some drawing. So here's our pre-mRNA. See, there's no AAAA there? What happens is there are these enzymes that they actually hitch a ride on RNA polymerase II. RNA pol II is this, like I mentioned, is this a ton of different protein subunits, and it has this long tail that's called the C-terminal domain. The C-terminal domain is like 10 times longer than RNA pol II itself. And I think of it as like a party boat for proteins. So that's not drawn to scale, clearly. RNA pol II, and this is like a tail, but it's referred to as the C-terminal domain, or the CTD. And what happens is there's a bunch of proteins that are hanging out that they basically hop off RNA pol II when they recognize their binding sites on the growing RNA transcript. There's a bunch of proteins involved, and again, they're listed here. I'm not holding you responsible for knowing the specific names of these proteins, but I do want you to understand that it's a group of proteins that are responsible for adding the poly A tail. Okay? So what happens is they're riding along, and they go, oh, snap, that's my binding site. And they hop off the CTD, and one of them, there's a cleavage stimulatory factor, stimulates cleavage of that transcript that RNA Pol II has been transcribing. So that's the first step, you get cleavage. And RNA Pol II is still like, oh, nothing, just transcribing along, and it falls off the transcript at some point. So RNA Pol II hasn't even stopped, it's still transcribing. So remember I talked about, there are 3 phases of transcription. Initiation, elongation, termination. This is the termination. It's the addition of the poly A tail that actually terminates transcription. So it's these proteins that were hanging out on RNA Pol II. They're like, oh, that's my binding site, that's my cue, take it off here. They hop off, cleave the transcript, and then another protein, poly A, polymerase, and there's a whole bunch of them, that then catalyze addition of the poly A tail. So you get cleavage, and then an enzyme that's like, cool, A, and on. That's very different than it being encoded in the genome, right? So one of my questions on the review questions is, what does that mean to not be encoded in the genome? What I mean by that is, no, it's rough to see art destroyed, isn't it? Okay. So here's our DNA. The poly A tail was encoded in the genome. Well, and here's our RNA. It's been transcribed. So it has A. What would you expect to see in the DNA? Yeah, or T, right? You'd see that at the end of every single gene. You do not see that. You do not see long stretches of T's, because RNA polymerase it's not making a copy of a stretch of T's that were encoded in the DNA to make a poly A tail. Does that make sense? There, right? We do not see this, right? The way this happens, the way the poly A tail happens is these proteins jump off Pol II onto the mRNA, right? This is an image of mRNA. Cleave it, and then add the poly A tail. So what's encoded in the genome is the binding site that recruits the factors that add the poly A tail. Binding sites are encoded in the genome, the poly A tail is not. Here is a bunch of the proteins that are involved in mammals. Again, there's no review question asking you, list all proteins involved. I'm not asking you to regurgitate that list of proteins on the test. I want you to understand this concept, right? Concept of not encoded in the genome. There are proteins that cleave the transcript and then add the poly A tail. Okay. So, some more features about the poly A tail that make it more confusing. Of course, is that a lot of genes have multiple sites where the poly A tail can potentially be added. And that can alter, if you choose a different poly A tail, poly A addition site, it can alter the way that gene is regulated. So here's where me showing you that the 3' UTR matters for translational regulation comes in. Okay. So in this image, what they're trying to show is the same mRNA getting a poly A tail added at 1, 2, 3, 4, 5. Did I just count that? I can count 5 extremely different sites. So I'm going to use an example to show how this can affect how expression of a gene might be regulated. Very well drawn. Okay. So, I guess I didn't need to, well, hopefully you guys got it.
Professor,
Yes.
so does that mean that changing a different site will alter the way the protein is regulated or functions?
It could be either. So that's a really great question, and it depends. So I'm going to talk about an example where the poly A tail site is just affecting the 3' UTR. UTR is not translated, right? Hence the name, untranslated region. But the way that translation is regulated, it can affect like the amount of protein that's made. But if you have a poly A site that's potentially in the middle of an exon and that gets chosen, now you're changing the part that's actually translated. If you change that, you're changing the primary sequence, changing the structure, you're potentially changing the function. Which is what those two, 1 and 2 are getting at, right? The alternative poly A site depends on where it's at. So first let's call, here's our, this is our 3' UTR. Okay, so this is actually a pretty poorly understood mechanism of why this happens. Here's one example where scientists have figured out what's going on at the molecular level. In a lot of cases, we don't know why one site is chosen instead of another. There are many questions yet to be investigated by budding scientists. Okay, so let's say we have a site here where we can have a translational inhibitor, right? So when this protein binds to this site right here, it inhibits translation, right? Okay. So we have, the way I've drawn it here is the poly A site would be in the case that I'm showing, so you'd have your 5' UTR over here. We're not talking about the 5' side, so we don't care. And again, this is not drawn accurately because really we would expect a circular conformation. Okay, so the case that we know a lot about are these in macrophages. Macrophages are a type of white blood cell, right, that are really important for our immune system function. So this is a super cool mechanism. So in macrophages equal white blood cell, important for immune response. Okay. So, in the absence of infection, this is kind of the scenario that you see, this poly A site selected over here. That you have a translational inhibitor binding. The macrophages don't need to be exhausting energy in translating defensin, which it's named because it provides defense against microbes, actually. So it helps our immune system respond to infection, bacterial infections, right? So what they found is this is my macrophage. Many bacteria release a molecule called LPS. And macrophages have receptors that can bind to LPS. Now remember when I talked about cell-cell communication, there's a ligand, finds a receptor, downstream response happens, right? And what you guys know so far is that downstream response can be anything in the cell. One of the things can be transcriptional regulation. So in this case, what LPS binding triggers expression of a protein involved in poly-A tail selection site. It was actually one of those ones on the list. Like, I just want you to know the concept, but it was, if any of you are like, which one? It's it leads to this upregulation of one of the cleavage stimulatory factors. And what happens when this protein is selected, it's when RNA Pol 2 is riding along, it's hanging out on the tail of RNA Pol 2, and it's like, no, this is my stop right here. And it gets off and causes a poly-A tail site to be added here instead. Okay, so if this transcript stops here, what do you lose? You lose the binding site of that translational inhibitor, right? So in this version, I have a sweet site in the 3' UTR that inhibits translation, so defensin doesn't get made. Here, we've evolved this mechanism to where our macrophages, they recognize that there's this bacterial-associated molecule in the environment, and it leads to the upregulation of a protein that says, oh, I'm going to select a different poly-A site. So you lose this whole part, right, of the UTR. So translational inhibitor is still there, but it doesn't have anything to bind to, so it can't inhibit translation. So now, all of a sudden, defensin is like, woohoo! And you get a ton of defensin protein made. And defensin can now help the macrophages kill the microbe. It's amazing.
Is that the same concept with the muscles being overly exerted, but they don't have a limit like that tough dog?
Whoa, what'd you just ask?
You know, like the tough dog where like they don't have that.
Oh, that's the buff dog, the whippet. That's a little bit different. I'm actually going to talk about the mechanism of how that happens later. It's an alternative splice site is chosen, so it's a little bit different. That's alternative splice site. This is alternative poly-A site. So there, yeah, you're affecting like the coding region. Here, you just lost half your UTR. And the reason that's relevant is because the other half had a site that inhibited translation. Okay, so there's a really long way of explaining, if you change the length of your UTR, you can alter translational regulation, right? Because you have all sorts of factors that combine at the UTR that can either promote translation or inhibit translation. You mess with them, you mess with how much translation happens. Yeah, question?
Since the grades exactly the same as ours, I'm really hard to choose.
Yeah. It is. This room is not ideal. Try to focus on cup half full. We have a room, so that's exciting. So, triggers expression of a protein involved in poly-A tail selection site.
And what's the parentheses above that?
Oh, CTSF. It's just the specific protein that gets expressed. So the slide here is the way I summarized it here. Right? So there are some bacterial compounds that increase expression of a protein in macrophages that selects a different poly-A site. So that's the short way to say in many words what I just said. Some bacterial compounds, LPS, increase expression of a protein in macrophages that selects a different poly-A site. Right, so that LPS binding the receptor triggered expression of a protein that was popped off RNA Pol 2 was like, I want this poly-A site, instead of the one that used to be on the board over here. In the absence of LPS, this protein isn't expressed, so you get the poly A site over here expressed. It inhibits translation of defenses. Basically, this is a way that the macrophages evolved a mechanism to make sure that we have that defensin transcript ready and waiting to be translated when it needs it. And the way that it gets it actually produced is LPS triggering the formation of an alternative poly A site. Okay. So if you're like, that's too much for me, what I really want you to understand is this concept, right? You change the length of the 3' UTR and can alter the translational regulation. The reason I like this is because it shows an actual mechanism that we understand how it happens, and it helps connect the dots to, oh, okay, you can have UTRs that translation is inhibited at a specific region of it. So if you lose that, it can promote translation. So that's how it can alter translational regulation. Okay. And what's perhaps a bit more intuitive is if there was a protein that was like, I want the poly A site to be right here. Now you're in the actual open reading frame of that protein. If you change the open reading frame, you can actually change the protein that's encoded. Yes?
These would be considered mutations, right? Because it's not affecting DNA?
Yeah, this is at the level of RNA. You got it. Yeah.
No mutation.
Mm-hmm. It's a change in the polyadenylation site. Listening? Okay. So functions of the 3' poly A tail. So obviously this is the last thing that happens. It terminates transcription. And the poly A tail, like the cap binding complex, has a bunch of proteins that bind to it too. They're called poly A binding proteins. Right, so it's nice. The proteins that bind the cap binding complex, the proteins that bind poly A tail, poly A binding proteins. And that helps indicate to the nuclear pore that this transcript is done being processed and it's ready to be exported. You kind of need all of that. There's a lot of proteins that bind along the entire stretch of the transcript, and they all help indicate to the nucleus this transcript is has been processed, ready to leave. Okay, so perhaps the most straightforward, right? It causes transcription to terminate. It promotes transport out of the nucleus. So it helps indicate, right, to these export factors, this transcript is done, right? That's the finishing touch, poly A. Regulates the initiation of translation. So its association, right, with the eukaryotic initiation factors, once they're recruited, that conformation is critical for translation beginning. So you need the poly A tail there, because that, along with the poly A binding proteins, bind the EIX. And that helps get translation initiated. And it also helps with degradation. But it's subtly different than the way that the cap does it. Right? So the cap has this conformation that isn't recognized by exonucleases. Poly A tail is recognized by exonucleases. So they start tuning it up. But it kind of functions as an hourglass. Right? So I mentioned that the tail can be different lengths. Right? So there is an exonuclease that starts tuning it up, but it goes at a certain rate. So if you have 1,000 adenosines, right? It's going to take a while to chew all the way in. If you only have 50, it's not going to take that long. So the transcript, right, once you get to the point where it reaches the transcript, actual open reading frame, the whole transcript gets degraded. There's a whole, it kind of, once the poly A tail is fully chewed up, it sets off a sequence of events that causes the whole transcript to be destroyed. So one of the ways the cell regulates how much translation do we want from this transcript is, if you only want a little, give it a short poly A tail. It's only going to be around for a little while. Because as long as the transcript's around, the translation machinery can translate off of it, right? You get multiple peptides translated off that one transcript. So if it's around for 5 minutes, maybe add a poly A tail like 50, cool, you get like whatever, 50 peptides, let's say. But if you have 1,000 adenosines, it's going to take a while. Maybe it persists for a few hours. Now you're going to get, let's just make it simple, 1,000 peptides, right? I'm totally making up these numbers. It's just to give you a sense of the longer the poly A tail, the longer that transcript's going to get to thwart the degradative mechanism, right, and get to be translated. Because most proteins, you want a certain amount translated, right? So how does the cell regulate it? This is one of the ways it regulates it, by giving it a shorter or longer poly A tail. Yeah.
So the proteins that they already have like a specific, like they have a specific poly A tail that's going to bind to that RNA. Like every time this one is going to be 1,000 because it needs to make a lot, and the other one is going to know it only needs 50 because it only wants to make 50.
That's a really great question. Yeah. So is it set for each transcript how long this poly A tail is going to be? And actually, that's a great segue into something I'm going to maybe get to today, depending on time. There are factors that can influence the length of the poly A tail. So again, this is kind of an active area of research where for some genes we know definitively the answer, like it kind of always has, a set length. But then for some proteins, there are extracellular factors that can affect the length of it. So just as a preview to what we'll talk about, a really cool example is a hormone called prolactin and the protein casein. Right, so prolactin is expressed, who knows what people express a lot of prolactin hormone? Breastfeeding women. What's casein? Milk protein. It's a very important milk protein for moms to produce nutritious milk for their pups or their babies that they're weaning, right? So what happens, part of what prolactin triggers is an extension in the poly A tail of the casein mRNA. So that's what helps moms produce way more protein. Yeah, it's amazing. Okay. All right. So RNA splicing. So this is probably the model of splicing that you learned about in Wave 4, I assume. Like you have exons and introns. Introns are removed to create kind of one continuous coding segment, and that's translated, right? And so at the boundaries of the intron, you have these consensus splice sites. They're recognized by spliceosome removed. Yeah? Is that about the gist of 184? Yeah? Okay. So we're going to build on that, and I'm going to talk to you about how that can vary from cell to cell. And that is called alternative splicing, right? Because it can create alternative proteins. Okay. So before you're like, what the heck is happening over here? Let's walk through kind of the key points. So first key point, alternative splicing is a way for a cell to produce a ton of different proteins from the same gene. Right? So this is why we can have 24,000 genes, and a water flea can have 34,000, and we're way more complex. We have way more proteins than a water flea, even though they technically have more genes than us. Because really, the functional molecule, the functional output, how many proteins do you have? That's what really matters. And we don't actually know how many proteins are in our proteome. It's really hard to predict. A bagillion, gazillion. No, I don't know. So the spliceosome, right, it's this big old complex of proteins and small nuclear RNA molecules. So to provide context by how big it is, guess how many proteins on average are in the human spliceosomes? What'd you say? 15. I like that. Do I get a 20? 25? 30? 200. 200. It is big. It is big. There are a lot of proteins. And so, right, when you have a big old protein machine, that means that allows for a lot of complexity, right? And so the spliceosome, it's not just big, well, it's not just big dumb scissors that is just like, oh, I'm going to cut here. There are factors that, that was my spliceosome impression, you're welcome. They can be different. They can be recruited differently to different sites. And that's where alternative splicing comes in. Okay. So alternative splice sites, when you have the same gene producing different protein products, they have that group of proteins produced by the same gene but are different only because they're spliced differently, they're called splicing isoforms. It's that specific terminology. So if you hear that proteins, that you have a group of splicing isoforms, that means that they were transcribed from the same gene and they were just spliced differently. Okay, so that brings us to these examples over here. So here is, this is representing mRNA from the BCL gene, and this is representing the mRNA from type 2 collagen. So these are both mRNA, right, or the pre-mRNA transcript. And you can see these two potential mature mRNAs that are produced. Those are going to produce different proteins. The proteins encoded by these different mature mRNAs patterns would be called splicing isoforms. Okay. So this mechanism allows for increased variability of genes without increasing the total number of genes. So let's look at these examples. Here we have, right, type 2 collagen, pre-mRNA, and there are 3 potential exons, right, 1, 2, and 3. And then here are these 2 in gray, the introns. It turns out that in precursor chondrocytes, so the teenager progenitor chondrocytes that are just finding their way, their spliceosomes recognize the boundaries that are precisely located at the boundaries of these 2 predicted introns. So what happens is those 2 get removed, right? The triangles represent those are removed, and you get mature mRNA composed of exon 1, 2, and 3. That whole thing's going to get translated. In mature chondrocytes, these splice sites are not recognized. Instead, the spliceosome just recognizes these boundaries here. So this entire region is removed. So in this case, you get mature mRNA transcript only composed of exons 1 and 3. So that's what's going to get translated. And that can alter the function of that protein, right? So the matrix, because collagen is important for ECM, this is going to change, right, the function of that collagen that's in the matrix, the extracellular matrix. So here's an example that's found in a number of breast cancers. So here we have the BCL gene, and in kind of normal tissue, what you see is the spliceosome recognizing these boundaries. So you get this intron removed, and then the spliceosome recognizing kind of a splice site that's in the middle of this protected exon. So you get a tiny exon 2. So in this case, it encodes a protein that promotes cell death. Right? So it promotes cell death in cells that need to undergo normal healthy apoptosis. In this case, this splice site isn't recognized. And so this whole exon remains. In this case, we create a BCL protein that inhibits cell death. Again, that can be really dangerous in malignant transformation because you have these cells that they should be triggered to undergo cell death. and they're not, right? That's part of malignant transformation is cells ignoring the rules of the body and not undergoing apoptosis formation. I always have a hard time when I have cancer examples because I know there's probably, based on statistics, some of you in here who know someone who's struggling with cancer right now. So I'm sorry if those are painful examples too. I have my stepfather died of pancreatic cancer 4 years ago, and it's still hard for me sometimes when I talk about how many students am I giving like, them an emotional experience right now, and then I'm like, okay, pay attention to molecular cell biology. Anyway, sorry, for life. I feel like I need to tell a funny story now that I went there. Oh my gosh, you guys. The day of your exam, that Monday, so I do these, like, when you become a mom, you're like, when are you going to work out? And so I work out before my kids wake up, right? And so sometimes I'm like, I'm going to get up at 4:30 and make their lunch first, and then I'll get to my exercise class. This on Monday, I was like, I just like roll. I always sleep in my gym clothes, just roll out of bed, and my gym's like 3 minutes from my house. So I've been awake for like all of 5 minutes, and I'm at this boot camp class, right? And all the boot camp classes, every time I'm like, am I actually going to do this? And then you do it, fine. But in the boot camp classes, there's like a line of treadmills, and then like you're going back and forth between treadmill and like crazy stuff on the floor. So I get there and I'm like half asleep, and the first thing you do is put your water bottle on the treadmill, right? I didn't notice that the treadmill that I was going to put my water bottle on was on. Like all the other ones were off, and so I like I step on it and instantly I'm like boom, boom, so loud, so loud. Yeah, it hurt really bad, and then of course like everyone is like, oh my gosh, are you okay? It's like, just my ego. Oh, it's awful. But you get up again, right? You just go. What the heck? The worst. I got thrown off the treadmill. Not my first time. So the other time I was thrown off the treadmill, I was actually sprinting, like full on sprint, and my foot hit the side, and the gym I was like, the windows were right behind me, so I hit the side and it was like, bam, and like threw me right into the windows, and that time was worse. Like this gym that I'm at now, I don't know, I think at 5:30 in the morning, it's all people who are like my age, and they're like, oh, are you okay? And then it was like, it was all, I don't know, people were like too cool. They're like, oh, this Asian person who just fell. Like, no one, like, people didn't even look at me. So, like, I'm leaving. So that's when I left. All right, so much about it. And it's life, right? It just happens. You get up, you go on. And plus, it's probably good for our egos to get hurt every once in a while, right? Like, humbling. So I'm telling myself. I got to make a lot of people laugh with the story. So. All right, so the thing that's really cool about this is, right, we have about, 22,000 to 24,000 genes. So do C. elegans, right? This model is this little tiny flatworm that's commonly used in science. They have about the same amount of genes, but only about 12% of their genes are spliced, whereas it's estimated 92 to 94% of our genes are spliced. So we can have only 24,000 genes, but have that encode, right, a bagillion, gazillion, or whatever I said, proteins. Right? This sky's kind of the limit. If you can have one gene that has the potential to encode 38,000 potential peptides. So you can kind of appreciate the variability that alternative splicing can provide. So the next few slides just have some examples of that. Right? So tropomyosin is one of these proteins that's pretty important for contractility. And you see different splicing isoforms in different cell types, right? So here's the mRNA, or the pre-mRNA, right, that's produced whenever tropomyosin is transcribed. It has the same pattern of exons and introns, but in these different cell types, it has different patterns of splicing that create different patterns of exons that are going to be translated. And what that means is, right, in smooth muscle is the only one that has this red exon. I don't know what happens, what the red exon does, but it has some function that's specific for smooth muscle, right, that tropomyosin isn't going to have in these other cell types. Right, so by splicing a transcript alternatively, you can get proteins with subtly different functions in these different cell types. So the one that has the, so far, the record is this gene DISCAM. And it got its name because it's associated with Down syndrome. So Down syndrome cell adhesion molecule. It's expressed in all of us. And it's really important in our neurons. So you know how before I mentioned that neurons have, one of their substructures is a dendrite, right? And the dendrites form these elaborate branching patterns. And one thing that you really don't want to have happen is any of those branches stick together. So what DISCAM does is it helps make sure that branches don't stick together inappropriately. And because we won't go into the details of the mechanism that I would love to talk to you at length in office hours about that if you would like, but it's really important to have different versions of DISCAM to make sure that all those dendrites that are in our brain, like we have 10 to the, I think, 11th power neurons, something like that, that have all these dendrites. You want to make sure they're not sticking together inappropriately. So you need a lot of variation in DISCAM to make sure that they do not. All right. Are you ready to learn about how alternative splicing happens? Yes, Professor Mulligan, it's the last 10 minutes of class when I'm always most attentive. Yeah, I know. It's 25-minute class with books. Excellent.
So having DISCAM is, it's associated with Down syndrome, you said, is it the lack of or the overexpression?
Well, in Down syndrome, right, you have, it's a really great office hour question, but I'll just say Down syndrome is called trisomy 21. They have an extra copy of it, and kind of all hell breaks loose. Yeah, you have all sorts of inappropriate wiring of the neural circuitry that happens when you have too much DISCAM around, basically. Yeah, it's awful. Okay, so here's basic look at what happens with the spliceosome, right? And I like that it's like cloud, other proteins, when I told you it's like 200, right? So you have this group of proteins and RNAs that come together to form these really complicated scissors. And one of the things the spliceosome does, right, is it removes introns. It recognizes the splice sites. It removes the introns. It helps bring together those exon boundaries. And I have a cool video that is going to be on the learning module. It's on your PowerPoint slides, but I'm putting that in the learning module that kind of shows this in action. It's really cool. And this cutout intron, right, it doesn't have a 5' cap. Exonucleases are like, yum, dinner. And the exonucleases eat that up. I was like this before I was an just so you guys know. There could be lots of things. This is just one thing. All right. So when the spliceosome leaves, it kind of leaves proteins there that stay associated with the RNA that also help lead it to the nuclear pore. And so those proteins are actually really important for governing where the spliceosome chooses to cut. And that's my segue to the next slide, which is, remember we had enhancers in DNA? There are also enhancers in RNA, splicing enhancers to say. And instead of binding transcription factors, they're binding recognition factors. So a splicing enhancer is RNA sequence that binds to a protein that's called a splicing recognition factor. So the splicing enhancer, RNA sequence. And again, because we're talking at the level of the primary RNA transcripts, they're all going to be the same. They're all going to have the same splicing enhancers, right? That's RNA, the primary RNA. And they can recruit or inhibit spliceosome formation. Right? So they can recognize those enhancers and say, spliceosome over here, this is where you cut, or they can be like, spliceosome, we don't want any. Go somewhere else. Now, the way that alternative splicing works is different cells, just like they have different combinations of transcription factors, can have different splicing recognition factors. So you can have in muscle cells specific recognition factors that bind to splicing enhancer sequences that make muscle-specific isoforms. Okay? And the way this kind of works is you have your exon, and then the splicing enhancer sequences at the exon intron boundary. So they can be, if the right recognition factor is there, cool, they'll bind it. If they're not, they won't. So I have a stripped down PowerPoint version of what this looks like, building off of this example of collagen 2 construct. Okay, so spliceosome bind to splice sites at the 5' and 3' end of introns, and they're recruited or blocked by these recognition factors. So a ton of different types of recognition factors. In my example, we have red and we have pink, right? So these represent 2 different recognition factors. Different combinations in different cells can lead to different differential splicing, different mRNA transcripts. Okay. So right in this example, we're going off of, right, we are in our precursor chondrocyte. So in this case, we have both pink and red. And in my example, we have red recognizing a splicing enhancer sequence out here. Pink recognizes splicing enhancer sequence here. They recruit spliceosome, and it cuts at these sites, removes the intron, and the gray, and you get the mature mRNA transcripts composed of exons 1, 2, and 3. All right. What if you lose pink? Right? So in my stripped down example, in our mature chondrocytes, I've drawn a scenario where you only have red. If you only have red, you only have recognition of these 2 sites. So in that case, big old spliceosome is going to be like, okay, cool, I'll just cut here and cut here. Right? And if you're just cutting here and here, you're removing that whole intervening region. Yes?
So are spliceosomes, are certain spliceosomes related to certain indicators or recognition factors?
All right, so the way that, so different people use kind of different language when they talk about spliceosomes. I feel like the most straightforward way to explain it is that the spliceosome just goes where the splicing recognition factors tell it to go. Right, so you have these scissors that I guess they are kind of big dumb scissors, and then the splicing recognition factor like over here.
So it's not like a certain indicator for different spliceosome sizes in the same?
So are there spliceosomes that are specifically recognized by specific recognition factors?
Yes.
Not that I'm aware of. I will look into it to see if there's any examples of that, however. This is kind of an area of science where it's been really active for a long time, and there's just still so many questions. Yeah, but yeah, I'll check it out. Yeah. Okay. And finally, just to wrap up, let's look at how this works with the myostatin gene, right? I talked about. So here's normal myostatin. There's 3 exons. The gray gets spliced out. So that mutation I talked about that leads to the non-functional myostatin, what happens is there's actually a mutation in the intron that creates a different splice site. So instead of the spliceosome taking this whole thing out, it just takes out a little part of the intron. So what happens is when it's being read by the ribosome, there's a termination codon right there. Usually that's removed, right? But instead, it just stops translating. So you get this truncated, non-functional protein. And right, so that is an example of a change in a splice site causing an alternative phenotype.